You can
Both. Very good.
See you.
Okay, so hello everybody. I'd like to begin by thanking Dr. Harikrishnan and Dr. Katta for inviting me to present our work on cardiac fibroblasts and to explain why and how cardiac fibroblasts need to be targeted to mend the breaking heart. I said the breaking heart, not the broken heart. Like other organs in the body, the heart is also made up of parenchyma and stroma. The parenchyma consists of terminally differentiated cardiac myocytes that are responsible for the contractile function of the heart. The stroma, on the other hand, has its cellular components, including cardiac fibroblasts, and an extracellular matrix scaffold. The quantitative relationship between parenchyma and stroma is extremely important for the structural and functional integrity of the heart. Surprisingly, cardiac function and dysfunction were viewed mainly in terms of cardiac myocytes, and the cardiac stroma was largely overlooked. However, pioneering work and extensive writing by Carl Weber turned the tide and put cardiac fibroblasts on the center stage alongside cardiac myocytes. Cardiac fibroblasts are cells of mesenchymal origin. They're the only intracardiac source of collagen types 1 and 3 that are major collagens in the heart. These cells also produce very many growth factors and cytokines that have profound paracrine effects on cardiac myocytes. In a setting of cardiac injury, normally quiescent or minimally active cardiac fibroblasts undergo phenotypic transformation into alpha smooth muscle actin expressing myofibroblasts that migrate to the site of injury, proliferate, and produce matrix proteins to form a collagen-rich scar that provides structural integrity to the heart. This completes the wound healing process. So far, so good. However, unlike active myofibroblasts in non-cardiac tissues that are eliminated by apoptosis once the healing response is completed, cardiac fibroblasts tend to resist apoptosis and persist in the infarct scar long after the healing response is completed. This leads to excessive collagen deposition, increased ventricular stiffness, and of course, diastolic dysfunction. Clearly, cardiac fibroblasts need to be targeted so that they do not overdo their job. The million-dollar question is, what in cardiac fibroblasts do we target? As we just saw, phenotypic transformation, proliferation, collagen production, and apoptosis resistance are key to cardiac fibroblast function in a setting of cardiac injury. Necessarily, cardiac fibroblast specific factors and mechanisms that target one or more of these processes would qualify to be a drug target. In this overall context, I must say that discoidin domain receptor 2 is a collagen receptor tyrosine kinase that has been implicated in a wide variety of fundamental cellular processes. And it has been found that several conditions, including tissue fibrosis, atherosclerosis, osteoarthritis, and so on, are associated with increased levels of DDR2. Now, in the heart, DDR2 is expressed predominantly in fibroblasts, which makes it a good candidate for targeting. Now, our studies on isolated cardiac fibroblasts, DDR2 null mice, spontaneously hypertensive rats, and a rhesus monkey model of metabolic syndrome point to DDR2 as a master switch in cardiac fibroblasts and an important determinant of tissue fibrosis. Now, how is DDR2 regulated in cardiac fibroblasts? All of us know that upon cardiac injury, there is a surge in intracardiac levels of angiotensin. And we find that angiotensin acts via the GPCR pathway to activate P38 MAP kinase that in turn activates the transcription factor NF-kappa B that binds to the DDR2 gene promoter to enhance its expression. Now, what are the implications of elevated DDR2 expression in cardiac fibroblasts? Using a combination of molecular approaches, we find that DDR2 has an obligate role in collagen expression, phenotypic transformation, proliferation or G1S transition, which is an important stage in cell proliferation, and apoptosis resistance in cardiac fibroblasts. Now, angiotensin is very well known to enhance collagen gene expression in cardiac fibroblasts. It also promotes the phenotypic transformation of cardiac fibroblasts, which is marked by the appearance of alpha smooth muscle actin. Now, we find that the stimulatory effect of angiotensin on collagen type 1 and alpha smooth muscle actin is either lost or significantly attenuated when DDR2 is knocked down using specific siRNA. Now, the mechanisms involved we have worked out, they're rather complex. We have, just like DDR2 is a collagen receptor, integrin beta 1 is another collagen receptor. Angiotensin enhances DDR2 as we have seen, and DDR2 promotes the expression, or rather enhances the expression of integrin beta 1 by a mechanism involving the AP1 transcription factor. It's a redox enzyme transcription factor. So in other words, we see a crosstalk between two collagen receptors here. And integrin beta 1 in turn enhances alpha smooth muscle actin expression, or it brings about phenotypic transformation. Now, a point to note here is that alpha smooth muscle actin is generally considered to be a mere cytoskeletal element. However, our studies show that it has an important role in collagen gene expression as well via the calcium channel TRPC6 and the YES activated protein or YAP transcription factor. So it's rather complex. So essentially it means that DDR2 integrin beta 1 crosstalk is a critical determinant of alpha smooth muscle actin dependent collagen expression in angiotensin stimulated cardiac fibroblasts. So from alpha, SMA and collagen, we move on to apoptosis resistance. We had reported earlier that the anti-apoptotic protein CIAP2 protects cardiac fibroblasts against ambient stress. Now, in this study, present study, we find that DDR2 knockdown, again using siRNA, abolishes angiotensin-stimulated IAP2 expression. Also, DDR2 overexpression in unstimulated cardiac fibroblasts induces the expression of IAP2, clearly indicating a role for DDR2 in IAP2 gene expression. Now, what does it mean in terms of resistance to ambient stress? We had shown that exposure of cardiac fibroblasts to oxidative stress enhances angiotensin production in these cells. And angiotensin, we have seen, enhances DDR2. DDR2 in turn activates the pathway shown here. The serum response factor, transcription factor is activated that binds to the IAP2 promoter and turns it on. And this confers apoptosis resistance because when you expose cells to oxidative stress after knocking down DDR2, there is an increase in apoptosis. So DDR2 does confer apoptosis resistance by a mechanism involving CIAP2 expression. Lastly, we also find that DDR2 promotes cardiac fibroblast hyperplasia or proliferation. Now, basically, again, adopting the same approach of overexpression and knockdown, we find that DDR2 knockdown in mitogen-stimulated cardiac fibroblasts produces cell cycle arrest at the G1S phase, at the G1 phase. These cells do not transit to the S phase where DNA is made, preparatory to cell division. They're arrested at the G1 phase. Similarly, when you overexpress DDR2 in mitogen in quiescent cells, those cells transit to the S phase. And these were shown by the RB retinoblastoma gene product phosphorylation status, pCNA levels, and of course, flow cytometry. So clearly, DDR2 has a role in cardiac fibroblast proliferation as well. Now, how does it do it? We have worked out the mechanisms thoroughly. It's far more complex than you would see here on this slide. Essentially, P27 is an inhibitor of the cell cycle. And P27 is regulated by factors like SKP2 and FOXO3A, which is a transcription factor of B27. Here, what happens is DDR2 activates ERK1/2 MAP kinase that inactivates FOXO3A, a transcription factor, leading to transcriptional repression of B27. Further, DDR2 also activates SRF, serum response factor, that enhances the expression of SKP2, which brings about the proteasomal degradation of P27. So in both instances, DDR2 removes P27. In one instance, by post-translational degradation of P27, and in the other, by transcriptional repression of P27. So in effect, by acting as a negative regulator of P27, DDR2 promotes cell cycle progression, G1 as transition in cardiac fibroblasts. So what we see here is DDR2-mediated ERK1/2 MAPT activation facilitates the coordinated regulation of cell survival and cell cycle progression in cardiac fibroblasts via serum response factor, because you found that SRF is involved even in apoptosis resistance. So it's a coordinated regulation by a single pathway. So to summarize our findings, experiments on isolated cardiac fibroblasts uncover DDR2-dependent molecular mechanisms that indicate an obligate role for DDR2 in cardiac fibroblast function. On to in vivo experiments, studies on DDR2 knockout mice indicate the link between DDR2 and collagen gene expression. Studies on spontaneously hypertensive rats point to an association, strong association between DDR2, phenotypic transformation, collagen expression, cell proliferation, and cell survival in a setting of myocardial fibrosis. Lastly, studies on a rhesus monkey model of metabolic syndrome that we carried out in collaboration with the NIH USA identify DDR2 as a molecular link between metabolic syndrome and arterial fibrosis. So how do you conclude? What conclusions do you arrive at based on all these? Well, our observations place phenotypic transformation, cell proliferation, collagen expression, and apoptosis resistance within a single mechanistic framework of interdependent processes under the regulatory control of DDR2 in cardiac fibroblasts. And here comes the DDR2 paradox. Paradoxically, by turning on survival mechanisms, DDR2 enables cardiac fibroblast-mediated wound repair in the short term, but it can potentially promote the persistence of these cells, leading to excessive fibrous tissue formation in the long run. Thus, cardiac fibroblasts and DDR2 are major contributors to the expansion of the cardiac ECM and consequent compromised ventricular compliance. They need to be targeted to protect the heart. As Providence would have it, the predominant localization and regulatory role of DDR2 in cardiac fibroblasts mark it as a potential drug target to control cardiac fibroblast-mediated cardiac fibrosis. So, target DDR2 and tame the fibroblasts to woo the heart. What are we up to? We are trying to have an in vivo model of coronary artery ligation and MI, in which, using a specific inhibitor of DDR2, we would like to see whether there is any functional improvement and the extent of fibrosis as well. And this is the team that has come up with all these. It started with Marina George, who is now in the US, Hari Krishnan, who is leaving for New York next month, Alan Sam Titus, who is also leaving for New York next month. I'm grateful to Dr. Edward G. Lakata, NIA, NIH, with whom I've had a very long association from 1997. Randy T. Cowling of the US UCSD is another collaborator. And of course, all this would not have been possible if not for the DBT, ICMR, and the Department of Science and Technology. So that is where we stand. A whole lot of data I've tried to compress in 10 minutes or so. Thank you.
Thank you, Shivakumar, for a very impressive presentation as always. One question, is there any physiological role for this molecule, and is there anything known about the temporal expression of this molecule during heart development?
Can you hear me?
Yes, yes.
Yeah.
Please.
One thing I didn't mention was, although DDR2 has been known to play a role in other fibroblasts and in other systems, DDR2 has not been studied in relation to the heart. First of all, as I said earlier, DDR2 is expressed predominantly in cardiac fibroblasts. Vascular smooth muscle cells and adventitial fibroblasts also expressed to a lesser extent though. But in relation to the heart, it has not been studied. So the field is wide open. So it could be doing it. One other thing we are not sure about, a fancy idea that you have, that we have, is DDR2 also is seen as a marker, a cardiac fibroblast, or at least a mesenchymal cell marker. So has it got to do with that? Something that is inherent to the cell type. You cannot be without it. I don't know. I mean, I'm not being negative on our own work, but I don't know whether that is the reason. In any case, it seems to be involved in very many things. The totality of cardiac fibroblast function, it seems to regulate.
Does it also have any regulatory role in arithmogenesis?
Again, I'll have to repeat what I told Professor Karta. It has not been studied, but the point is, since it regulates collagen, and since collagen is known to interfere, the scar is known to interfere with rhythm, probably it would. I mean, at least indirectly, if not directly. I mean, that's another issue that we need to look at.
Okay, because of time, I think we will move to Dr. Shakti.